SolveRD, Solving the Unsolved Rare Diseases, is a youth-funded research project funded for 5 years, and we started on 1st January last year. So we have the first year just passed, and we have 4 years to go. We have 22 partners from 10 countries in Europe and the US. And this project is coordinated by Olaf Ries and Holm Gressner here from Tübingen, and co-coordinated by Han Brunner and Tony Brooks. SolveRD builds on existing resources and infrastructures. So we have a group of 4 core ERNs, the European Reference Networks in SolveRD, which is the ERN R&D, EuroNMD, Ithaca, and Genturis. And we have already associated a number of more partners and several networks, so 6 additional ERNs, for example, and 3 undiagnosed disease programs. We build on existing infrastructures like RD Connect and Elixir, Orphanet, Human Phenotype Ontology, Eurogentis, or the Canadian Models and Mechanisms Network. And last but not least, we also have patient organizations on board, which are Eurodis and Genetic Alliance UK. As I said, SolveRD is about solving the unsolved rare diseases. And to achieve this, we have defined 4 different cohorts. The first cohort is the unsolved cases. So patients and their family members with an inconclusive exome or genome. And those cases will be reanalyzed using standard analysis pipelines of GPAP. The second is the specific ERN cohorts. They are defined by our 4 core ERNs, and they will receive all kind of omics analysis depending on the cohort. The third cohort are ultra-rare diseases, again defined by the 4 core ERNs, and those are phenotypically most special or remarkable patients with a rare disease without an exome. And then finally, the 4th cohort, the unsolvables, defined by our core ERNs, highly recognizable clinically defined diseases or syndromes, for which no disease gene was identified yet, despite exome or genome sequencing and additional considerable research investigated. But here we would like to focus on the first cohort, the unsolved cases, because we are currently collecting these unsolved cases from our ERN partners, associated partners, and associated networks. And for this, we need a PhenoTips entry, and of course, the exome or genome, which has been done. And how we collect this and how we do PhenoTips entries is being explained by Leslie. Okay. So I now hand over to Leslie. Leslie, could you please share your screen?
Yes. Can you hear me well?
Yes. But we cannot see you yet. Well.
Okay. I share my desktop. Can you see my slides now?
Yes.
Yeah, we can see that.
Perfect. So I'm Leslie Matalonga. I'm here together with Stephen Lowry and Ricky Yoshi from the SolveRD project, and we are all from the Centro Nacional de Análisis Genómico here in Barcelona. So today this webinar is to show you how to submit phenotypic and genomic data to the RD Connect Genome-Phenome analysis platform. So first of all, the data workflow within the SolveRD and in connection with the RD Connect GPAP has 5 different steps. The first one would be the registration, and I will quickly show you later where you have to do this step. The second one would be to submit the phenotypic data of all the individuals you would like to reanalyze and submit also genomic data. And for this, we will have 2 different options: to bulk upload the phenotypic data if it is already HPO or MIM ordo encoded, or to enter this phenotypic data using our PhenoTips instance and creating individual entries. It's important to know that you will need to create entries for the index cases, but also for all the relatives that you will include and submit genomic data for. In all the steps, you will find guidelines. I will show you later where are the links, and also videos that can help you in the process. Also to mention at this stage that within SolveRD, you can submit your unsolved cases, but also your solved cases. So once you've done this second step, submitting the phenotypic data, you will submit the genomic data that belongs to those patients. Again, I will show you that we have detailed guidelines that can help you on this. And here we will see that there are also two options: to bulk upload if you have many samples, or to use user-friendly online tables on our system. So finally, once you've submit the phenotypic and genomic data, we will process this data, and you will be able to reanalyze it through the Genome-Phenome analysis platform. We have some tutorials and videos for this to show you how it works. Finally, once you have a reanalyzed data, then if the case has been solved, you will need to tag the variant in our system. Or if the case is still unsolved, then there are different approaches like patient matchmaking, or they will go through the SolveRD data analysis task force. So in today's webinar, we will explain how to submit this phenotypic and genomic data to the RD Connect GPAP. And on Friday, Stephen Lowry will be giving a webinar on how to reanalyze your data within the GPAP. If you have any question, please use the chat box. Ricky will be managing this, and we will answer all of them, or we'll try to at the end of this webinar. So this is the RD Connect landing page. Here you will find all the links to the different parts I just presented. So for the registration, the GPAP and the submission, and for our Phenotypic instance. So for the registration, you will just need to click in the link in this landing page. And you will need to fill in and complete the registration process, which has 3 easy steps. This only is required by the PI of the group, and the PI will also include all the team members and notifying the emails that have to be registered. You will have to provide a photo of an ID and also to sign the adherence agreement. And after 2 to 5 days, the Data Access Committee will approve your request, and you will be sent credentials to log in the GPAP. Also, just here to mention that as you are part of SolveRD, you will have to indicate this in this registration so that we can record this information. Okay, so now the second step, once you have your credentials to access the platform, would be to submit the phenotypic data. So as I mentioned before, here you will have 2 possibilities. One is to bulk upload your data, and this is possible only if your data is already HPO or MIM or ORDO encoded, and it's already available in a specific database or file on our site, on your site. And it's strongly recommended if you have a high number of patients, it will be easier. The second option is to use user-friendly online templates that have been designed in collaboration with the ERMs. And this is useful when your data is not already encoded with ontologies, or also if you have few number of patients to submit, as it's really user-friendly and quite fast. For both approaches, you will need to enter a minimum number of fields which are required within the SolveRD project for index cases. So these mandatory fields are the sex, the affected status, the date of birth, or at least the year, also to add information about the pedigree. And finally, we also request to submit at least 5 HPO terms, so 5 symptoms, if possible, associated to your index cases. Okay, now I will show you how to perform the bulk upload in case you already have your phenotypic data encoded in OMIM, HPO, and ORDO codes. So if your data is already in another phenotype instance, then you will just need to export this data and send it to us. We will import it into our RD Connect phenotypes instance, and we will send you the corresponding IDs. The second option, if you have your data already HPO, MIM, and ORDO encoded, but not in a phenotypes instance, so in your own database, In this case, what you will need to do is to download an Excel template that you can find in the phenotypes guidelines in step 4.1, and to transfer your information into this Excel file. Then we will convert it in a format that can be directly imported into phenotypes, and we will send you the corresponding IDs. In this Excel file, you will find mandatory fields in red, the ones that I mentioned before also. And you will also need to enter information about the index cases and any relative you would like to submit genomic data. In the second row, you will find information about the information which is expected in the column. And also, as I mentioned before, you will need to enter the HPOs and at least 5 HPOs if possible. Here I have also added the link to the browser, the HPO browser, so that you can find the corresponding codes if you want to. Also, I would like to mention that QC verification will be performed once we have imported the data in the system, and we will check if the minimum phenotypic information required has been submitted, and if it's missing, we might request you to add this information. The second option is to use these user-friendly online templates that have been designed with, for now, the 4 core ends in the project. This will be useful if your phenotypic data is not yet structured using the ontologies. For now, we have this list of disorders associated to each of the ends, and this will enable you to enter the data in the system directly. For each of these templates, you will find a list of HPOs that are tailored for each of the different disorders. If you cannot find any disorder that fits your patient, then you can use a SolveRD minimum field default form that I will show you later how to select it. And there you can enter any type of disorder or patient. Okay, now I will show you a little demo on how to enter this phenotypic information directly in our system. I will use an example. So a 14-year-old male presenting with muscular dystrophy. Symptoms are swallowing problems, respiratory insufficiency. He doesn't present cardiomyopathy. And also we have laboratory results on serum creatine kinase, which is elevated, or absence of dystrophin. So now, okay, this is how phenotypes looks like. You will access this page from here, the landing page, just clicking in Access Phenotypes. Then you will enter your credentials. And on the right, you will find a list of all your cases and your patients. So to create a new one, I would go here to Create and select New Patient. And here is where we will find all the different templates that are available. Here is the SolveRD minimum field if none of these templates fits the clinical symptoms of your patient. In our case, we will select muscular dystrophy, and I click on Create. We strongly recommend to always start entering the information of the index case, and from there entering more relatives. So the first thing I would do is to enter my local identifier to always know to which patient this phenotype entry corresponds. So here I can enter my patient webinar, for example. whatever ID, local ID you want for the patient. Then I would enter it's a male, alive, and also this is an important field, which is the clinical status. It's mandatory, and you need to tell us if this patient is affected. So I would select yes, or if it's unaffected, I would click on no. In this case, I'm entering the index case, so it's affected. Here I would enter the date of birth, and only the year is mandatory. And then you can go through the form and fill in the corresponding information. You will see that there are different fields. If you need any information on what is required, you have this information that you can click there and have a little description on this HPO. So here we enter the global age of onset and the progression is a progressive disorder. So once I have entered a little bit of information about the case, we will draw the pedigree. This is also a mandatory field, and it's our only way to link the family and all the relatives that you will submit. So I would click here on Draw Pedigree, and I will create a new family. So there is a pedigree editor embedded into PhenoTips, and here I can select different templates. If you only have an index case, you would select the first one. In this case, I would enter a trio. So I will select the second one. The information I have already entered is present here in my index case. I can add more information if I click on the case. I can add more personal information or clinical information. I can click here on affected to show that, quickly see after in my pedigree who are the affected individuals. And yes, you can complete all the information you think might be relevant for the analysis. Then if I have genomic data on any of the other relatives, I will need to create another Phenotypic entry. And we strongly recommend to do it from the pedigree. So you would click, for example, in the mother, and to add this new entry, I will select Create New. So here just an advertisement to tell me that it will be linked to this family, so I confirm. And now I have this new Phenotypic entry. So here, if I click, I will go to the entry of the mother, and I can edit it and complete all the relevant information. I will stay in my pedigree, and what I would do is also to create the Phenotypic entry for the father. So I create the new confirm. Okay, so in this case, if I have genomic data from these 3 patients, I have created 3 different Phenotypic entries, which are linked to my family through this pedigree. So I would save the pedigree, and now close, and this will bring me back to my index case form. Now you can see that we have our pedigree in our form. Then I will add information about the global mode of inheritance. Again, you have this information if you want more detailed description of each of the fields. Here I will select sporadic. And the next step is to enter the clinical symptoms of the patient. In the case of the parents, I would select here, this patient is clinically normal as they don't present any symptoms. As now we are entering the index case, I will show you how to enter the phenotypic data. As we have selected muscular dystrophy, what I have here is the typical symptom that this type of patients might present. So what I can do is to quickly browse these trees, selecting different HPOs, and enter the information. For example, we know that our case has swallowing problems, so I can select directly from here that yes, this symptom is present. You will see that these arrows give you a more detailed HPO term. If I find a more detailed one, then we will ask you to select the more detailed one and remove the one upper in the tree, so that we are not duplicating the same phenotypic information. In this case, I will keep the swallowing problems. You can keep going through the tree and, for example, go to respiratory dysfunction, and here we will find the respiratory dysfunction HPO. I will select this one. Also, if I continue, there is no cardiomyopathy, so I would here select that this symptom is not present. And as you can see, as I am adding this information on my right part of the screen, all the HPOs I'm selecting are being added. And at the top, you have an indication of the quality of the phenotypic data you are entering. If I cannot find my symptoms in these trees, I can also use the browser on the top. For example, in this case, if I want to enter that there is no dystrophin in the biopsy, I would start typing, and you will see that the different HPOs available matching the information you are trying to access will appear. So in this case, I would select absent muscle dystrophin expression. And I can do the same with the creatine kinase deficiency, abnormal creatine kinase levels. And you see that as I'm entering the information, the quality is being updated. So we recommend, if you can be between good and very good phenotypic information, it will be better for the analysis of the genomic data. So once you have entered all your phenotypic information, then there are just a few steps more. The diagnosis to enter the ordo, the clinical disease with an ordo code. Here it's also a browser, so I would start typing my muscular dystrophy. And yeah, and you can select it from the drop down, and you will see that it will automatically link to an ordo code. If I'm submitting a solved case, then I would also add the OMIM entry. So if I know that my case is a Duchenne dystrophy, I can also add this information so it's recorded in the system. Then you can add genomic information if previous studies has already been performed, and you can add if you have, for example, screen a specific gene and it has been negative, or if there is a candidate, or if it's the confirmed causal. So this information is also recorded. And finally, in this last step, the case resolution, you would indicate if this case is solved or not. So if you are already submitting a solved case, you will have to check this box, or if after reanalyzing it through the GPAP that Steve will show you on Friday, you solve your case, you will have to go back here and click this box. Okay, so once you have entered all this information, you will save and view summary. So you can see all the information you have entered. Okay, to be able to submit genomic data, what you will need to do is to share this case with your group. So you will need, after entering the phenotypic information, to go here to modify permissions. And at the bottom, you will see new collaborating group. So here you will have to enter the name of your group. In my case, I entered the SINAC, and I'm sharing it with all my SINAC group. And then I update the record, and this is now here recorded. If you want more information or something is not clear, we have the Phenotypics guidelines, which are here, and also you can find the link in the RD Connect database. So from here, you can click on any of these steps and review the information that is required. Okay, so in summary, you can submit unsolved and solved cases. You will need to create an individual entry for all index cases and the relatives you are going to submit genomic data. You have two options for submit this data through bulk upload and the Excel files that I've shown, or directly exporting from other Phenotypics entries, or using these individual forms through Phenotypics. And in this case, remember that you will need to share with your group all the individual entries you will submit genomic data. Here is the link to more detailed guidelines. And of course, if you have any question, you can contact us at this email address. Okay, so once you have submitted your phenotypic data, then you will be able to submit the corresponding genomic data. So for this, you will go to submit data in the RD Connect landing page. And also, if you need, on the right, you will find the guidelines. Again, here you have two options: to bulk upload the data to the system, also using Excel files, I will show you later, and or using online tables on our system. So here is how our interface looks like. You have two parts in this data submission module. The first one, and the one that we will be explaining today, is to submit the data to RD Connect. And the second one, the Manage Your RD Connect, is for data that is already in the system, so that you can see all your sample, experiments, and all the information associated. So the first part is useful because you will link the genomic data to the phenotypic data you have already entered. And you will also enter at this stage all the description of those data sets, which is the metadata. These data sets will also be associated with the RD project and be shared with all the members of this project. And also you will share it with all people from your ERN. Once you have entered all these metadata or description of your data set, you will receive an email that will give you the link to upload the raw genomic files. We use an Aspera server. I will show you later how to do it, but it's very user-friendly, and you will just need to enter the files and no more information, as all the metadata will already be in our system. The second part, I won't enter into details, but it will enable you to modify sharing permission, to enable patients to be matchable through Matchmaker Exchange, for example, to export all your metadata, to track which users have accessed your data, etc. So now I will show you how to submit data from a trio. So for example, the trio we have just entered in the system. So we'll need to follow three different steps. The first one will be the participant step, where we will give to the system the phenotypes IDs for the patient we are going to submit experiments. The second one will be to enter the information about which experiments we are going to submit, so exomes and which kits. And the third one will be to tell the system the name of the files we are going to submit, so that we can match everything and process all the information. Once you complete these three steps, then as I said before, you will receive an email with instruction on how to use Aspera to send us your files. Okay. So I will access, as I said, the submission through the landing page here on Submit Data. And I already have it. Okay. And this is the page. I would select first go to Submit Participant. And here I will find the two options, the bulk upload and the online tables. I can start filling in the online tables, which is this part. So I would select my phenotypes ID from the list. This is all the phenotypes ID that my group owns. So I will select one phenotypes ID. For all the fields that you have also question marks, and if you hover the mouse, it will give you more information about what we are expecting. And if it's a mandatory field, you will see a red asterisk. So here I would enter the phenotypes ID and check if I want my patient to be matchable or not. This is one possibility. The other one If I have more patients and I want to do the bulk upload, I will need to download this Excel template. So I would click here, and it will download my template. I already have it there. Here you will find in the first table the same information that I've shown before. In red, you will have the mandatory fields. And if you want more details, you can also go to the second tab and find more information and description of any of those fields. So here I would enter my phenotypes ID and information. Generally, you will see that there are drop-downs about the different fields. To submit this data, I need to save this file in a TSV format. You also have detailed information in the guidelines. It's not difficult, but you will need to go to Save As and select, instead of Excel, you will see that the different systems somehow have a tab delimited format. So you will need to use this one to save your file. Okay, so once your file is saved, I won't do it there. Now what I can do is to browse and enter this file into the system. So I would browse, select my TSV file, this one, step 1 participant. Open, and you will see that the information will be automatically filling in the system. So once I have everything here, I will just click to submit. And this has submitted all my information from step 1. Here you can enter as many phenotypes IDs as you want. And then we will go to the second step. We have a link directly here to go to submit experiment. And again, here we have 2 different approaches. To fill in, if you have few samples, all the information using the tables, or to download the Excel template and fill the information through this Excel. So here I also have, for example, pre-fill our Excel file with the information in red, the one that is mandatory, and also using drop-downs when available. Just to mention that in the second step of experiment, you will need to select the ERN you belong to, and here you will have all the ERNs listed. And also you will need to enter the project. So here your sample will belong to the RD project. So again, to enter this information, I would browse my file that I have saved in a TSV format, and I enter my step 2 experiment. The information is automatically filled in, and I will submit the query. Here is a summary of what metadata I have been submitting. And now I can go to step 3 to say that for each of these experiments, so here I submitted an exome for each of my patients. Now I have to tell which files will be associated to this exome. So this is the third step. Again, here you can manually enter the information. In this drop-down, you will find the local IDs you have entered, and on the right, you will find which RD Connect ID has been associated. But this you will be able also to export it once finished. And from here, you have to enter which files will be submitted. So if I have my FASTQ files in a folder, what I would say is for each of my experiments, select which files will be submitted. At this stage, you are not submitting that file, but you are just giving us the name of the file so that we will be able to match it afterwards. So here, for my first experiment, if I have 2 files, I would select both and submit them. So now we have registered this name. I would select my experiment first, for example, this one, test 1, and add the information. Again, the one with a red asterisk are the mandatory ones. different strategy, and yeah. So in this case, now I have all the information. If I want to use an Excel file, I would do the same. For each of my experiments, enter the name, the exact name of the file, and then fill the information. And I save a TSV, and I can browse this file into the system. The information is automatically filled in, and now I won't do it, but because then I'm submitting those files to the system, but you would click on submit here. Okay, so once we submit this first step, you will automatically receive, as we said before, an email with credentials to use the Aspera server to really send us those FASTQ files. So the Aspera server, we use RD for providing this server, and it's a really secure, robust, and quick server. It's 10 times faster than the FTP. That's why we are using it. And in case you are interested, it can also be accessed by command line. So you will be given credentials, and you will access the link we provide. And once you are there, you will just need to click here in Upload Files, and then send us all the files you have entered the metadata in the submission module. As you have already told us in the third step, which is the name of those files, we already have all the information to associate all these files with their corresponding experiments and their corresponding phenotypes IDs. Okay, so now just a quick summary of what we've seen. So before starting the submission, you have to ensure that you have created and share with your group the corresponding phenotypes entries. Also ensure that before starting step 3, you have the name of the exact name of the file that you are going to submit. Again, you have two options: to bulk upload using those Excels, or to enter the information through the online template. You will find also detailed guidelines explaining each of these steps. And again, of course, if you have any questions or doubt, you can contact us at this email. Okay, so now we've explained how to submit the phenotypic and the genomic data, and on Friday, Steve will show you how to analyze when we have processed all this data. So yeah, thank you for your attention, and now we have some time for questions. Yes, we have almost 20 minutes for questions. I think Ricky has been tracking the question in the chat, so we are going to try to answer them. And if you have more questions, just write them down in the chat as we are monitoring it. So Ricky.
So there's a few questions that have been sent. So please, if you have any questions, write them into the chat box. Please make sure that it's addressed to everyone. You'll find the chat at the bottom of your screen. So the first question is by Sophia. Is it necessary to draw a pedigree, or can we just submit the possible inheritance patterns? Because she thinks that it could be too time-consuming and it could dissuade her from submitting more cases.
Okay, it is necessary to draw the pedigree because it's our only way to link the family members. But if you are submitting only index cases, you've seen that there is a template for an index case. So you can just click on draw pedigree, select that template, and then you don't have to do anything else. It's just save these, and so it won't take time.
And I would just add that you only need to include the people in the pedigree for whom you're submitting the genomic data. So if you have to go to if you have a sample and a cousin, you would not need to fill in it. You would not need to make phenotypes entries necessarily for the parents or the uncles if you don't have them. You would just make a phenotypes entry for the index case and her cousin. But when you draw the pedigree tree, you would obviously go up to grandparents and back down, but you would only have 2 phenotypes entries.
Yeah. And also think that this information is important because well, it can be useful for the analysis, and if you have other affected members in the family, it's always useful to know it.
Okay. Yeah. A question by Sunita. If you have siblings, how do you add the siblings to the pedigree?
I think Leslie can maybe show it in the phenotype entry she made with the trio.
Yeah.
She'll show you how you can actually extend the template. So the template is only to get you started with your phenotypes entry. Afterwards, you can extend it as much as you need to. So.
Yeah, sorry, I haven't shown this. So here, for example, we have our pedigree, and I can add siblings, for example, clicking here. And if we have a sister, I would select a female, and then you will see that well, the sibling is created. And you can go up on the tree as far as you want if you have the information, or the same if you have any children that you want to associate to your index case. So yeah, browsing the pedigree, you will see that you have many options to enter all the information.
Okay, yeah. A question by Spencer. Could the template be used to integrate a decision tree structure to help the diagnosis? So when making a new template from scratch, I think it was just talking about how it would help the clinicians.
You mean, well, the question means to create a new template for an ERN that can help, then yes. In this case, I would propose you to contact us, and we will see how we can improve those templates. We are very happy to do it, and if you have other HPOs to propose that you think might be more useful, then of course we can discuss this.
A question by the team in TIGEM is one of the HPO terms was marked as obsolete. Is that an issue?
We update periodically our HPO, the HPO file we are using for phenotypes. It might happen, but we'll try to solve this in the future, but it shouldn't happen very often. We'll try to update the file as frequently as possible.
I'm not sure if that meant if it was obsolete
in the ontology.
In the ontology, or obsolete within phenotypes. So
because the ontology updates, so sometimes it's true that we are not in the last update because we use a file for phenotypes, but normally we try to update these files every 2 months or 3 months.
So in this case, I would recommend that if you find that it's not in our phenotypes, or it is and it's obsolete, then you could let us know. In other words, if you can find the term that has replaced it, check if that's already in our phenotypes, because it might be that there was a duplication of term of some sort in HPO, and then obviously they make one of the duplications obsolete, so the other term is the correct one.
Yeah. And a question from Berta. In phenotypes, at the top of the page, there are 2 checkboxes. One is for the patient consent. So are these checkboxes, are they mandatory fields?
No, in fact, we are going to remove those checkboxes as this kind of information is already taken into account within the Solve-RD project, and you don't need to check it in phenotypes at this stage. This is why I haven't mentioned them.
There's a comment by Anna who suggests that if it's possible to have a system whereby if the phenotypes entry is not shared properly or hasn't been saved, if a note can come up to tell you that that it's not been basically the information hasn't been entered properly.
Phenotypes automatically save every, I think, 30 seconds or something like that. So you might not lose all the information entered, but no, at this point there is no warning if you are leaving without saving. But if you do so, you might be able to recover quite a grand part of your information.
Okay, Amana's just commented, not the saving, the sharing.
If you forgot to share, then what will happen is that in the submission, you won't see your phenotypes entry in the list. You can go back to phenotypes, and as I said in the main page, you will have a list of all your patients. So you can just to go back, select it, and share it. Or you have a browser here where you can also enter the phenotypes ID, and you go back and edit the form. So there is no problem at all if you forgot to do this at the moment of entering the phenotypic data.
The thing is, Anna, that we can't assume that you want to share the data. So there's no way of us giving you a warning that you haven't shared, unfortunately, because some people do not want to share the data.
Okay.
When I say some people, sorry, I mean some people who use PhenoTips. So this is a PhenoTips feature, not a SolveID feature.
Okay, so I hope we're answering your questions. If not, please let us know. I will be more than happy to discuss. There's another question that is related to the pedigree question. How do you get the pedigrees for bulk upload?
In the bulk upload, we ask you to enter the who are the relatives, but at this stage we are not able to draw the pedigree. What we will have is in our back end the information about that relatives are from the same family, but we might ask you to enter the phenotypes instances and to link those entries within the pedigree.
So there's a question by Enzo, which is a question that we get asked quite often, is how they should fill in the minimum of 5 HPO terms required for the phenotypes entry in the case of unaffected relatives.
Yeah, sorry, so maybe I haven't been clear. These minimal requirements are for the index cases, either for unaffected relatives or other relatives that might be affected. Of course, if they are affected, we also would like to have this information because it's important for the analysis, but for unaffected relatives, you don't have to add any HPO.
We have another question by Tigem. Is there a minimum level of information for the non-affected relatives phenotypes?
Yes, well, what we ask is if you can enter the year of birth and also the affected status, at least for the relatives.
And there's a question, this is at the moment our last question, is if you have a patient, for example, with a non-syndromic intellectual disability, 5 HPO terms is pretty complicated. What would the user do in this?
Yes, we know that some types of disorder, also with cancer risk tumors, risk tumors, it might be complicated to have 5 HPOs. In some cases, we can make an exception, but you still have to think that it's very important to enter deep phenotyping for next steps to try to solve the cases. So please just enter less HPOs if it's really because there are no more phenotypic description associated. We are also in this project, Sebastian Kohler is also, he is working in the HPO ontology, and if you think that it's just because the term is not there, then please contact us, and we are in contact with him, and he will be very happy to update the ontology if necessary.
Okay, so going back to the HPO terms, there's a question also from Tigam that says, if the parents do not need HPOs, how will we submit the parent information?
In the same, if it's a bulk upload in the Excel, you will need to enter this a row with the the ID and state the relatedness to the index case, if it's a father, mother, etc. But then you don't have to enter any HPO at this stage. And if you are using the user interface, you have to create the phenotypes entry, but you don't need to edit it and add any HPO. You will just state that the patient is clinically normal. And remember that in all the cases, at the beginning, you will have to say if it's an affected or non-affected relative.
Okay, yeah, nice. And the question by Steve, what should I do if I make an error when submitting data, phenotypic or metadata?
For the phenotypic data, you can always edit your form at any stage. Also, if you have, imagine more information later, you can add it to the system. And there is a history of all the edits that have been made. In the submission, you will need to contact us. Yeah, the help, you have the email help at rdconnect.eu, and then we will modify any experiment or any metadata that needs to be changed. We don't allow to delete experiments by yourself because it might be problematic for us for tracking purposes. So this is why for the metadata, we ask you to contact us.
I have another question is, so what happened when the case becomes solved?
So if you solve a case in the GPAP, and tomorrow Steve will show you how to analyze the data, then you will need to go back to Phenotypic and enter the OMIM code associated to this disorder if existing. And at the bottom, I don't know if, I don't have it now here, but at the bottom, I don't know if you remember, there is the case resolution status, and you will need to just check this box. And then we will know, and it will be recorded for the Solvardi project, that this case has been solved. It's very important to do this step. Of course, you can always access the guidelines, and this is explained here in 8.1 solve cases. So once you solve a case, here you will have detailed guidelines on what you should do in the genomic and the phenotypes instance that I've just explained.
Cool. The last question is, why do we need 5 HPO terms?
This was agreed at the beginning of the project because, for example, work package 1 needs phenotypic information to try to find through algorithm possible disorders or to describe patients with possible new disease entities. And we found, and expert clinicians found, that having 5 HPO was enough to be able to do these things. So this is why less than 5 HPO might be a problem for them, and instead of requiring more information later, we would like to have this information at the beginning. And also because, as you will see tomorrow, we integrate phenotypic information within the analysis. So if we have a detailed phenotypic description, you will see that it will enable you to filter your genomic data and try to find the causative variant. And it's really an added value for this. It's not just to have on a database. We really use this phenotypic information as in RD Connect, but also in the whole Solvardi project. And for the data analysis task force, and well, you know.
Okay, we have another question from Tikem that says that if they send us a JSON file that's downloaded from Phenome Central and there's only index cases, will we be able to add the parent information?
Yes. We will first import this information into our phenotypes instance and give you the corresponding phenotypes IDs. And then you will have 2 options to enter relative information. Use the bulk upload and just tell us who's the index case and the phenotypes ID, and then enter the information of the relatives. Or if you have few cases, then go in the phenotypes instance and from the pedigree link, create the entries from the relatives and link to the index case.
Okay. So we've had also two questions that have actually been answered within the chat, but I'll reveal them anyway, just in case you haven't seen them, because I think they're quite relevant. One was by Sophia, who asked if we have to fill in the absence or the signs or the symptoms. And Anna, thank you very much for replying to that. She wrote, in my experience, it is important to add the negative signs only when they have clinical relevance, such as absence of cardiomyopathy in an MD case. This is exactly the answer. So thank you very much for that, Anna. And Sophia also asked about the logging question. So if you get confirmation of the logging details, and if they get added to the registered project PI. And Steve, thank you very much for answering that. And indeed, what he said is exactly the answer. So the user should receive a username and a password. The username is the same for Phenotypics and the Genomics platform, but the passwords will be different. The way that you actually get access to both Phenotypics and the GPAP is that the PI has to give us a confirmation to add the user to the group. And with this, every user will get a user created within both of the groups, and you'll also receive details on exactly what you need to do to activate your user details and your account. Until you have this, you won't be able to access nor Phenotypics nor the GPAP. So we have another couple of questions that have come through. One from Sophia, where she says, where she's talking about the minimum of the absent signs or the symptoms to fill in, or it just makes part of the 5 HPO term rule. So
normally we expect 5 positive HPOs and then any relevant negative one. Generally, the rule is for positive HPOs, because these are the ones that can really be used for analyzing the genomic data.
And if all my cases have exactly the same HPOs, because that's what defines the disease, do I need to enter anything?
You should enter all the HPOs for all of your different index cases, even if they are the same, because it's important to remember that we are all in a very huge project, so maybe your cases might be interesting for another, and this phenotypic description for another group, so it's very important to always submit everything.
Okay, so it seems as though we don't have any more questions. If you do, please quickly write them. If not, oh, here's one from Tigam, which says, the disease template is a custom feature of your Phenotypics instance, or could it be used also in another instance?
It's specific from our instance. Yeah. And created within the Solvardi project, updated at least, was already used for Neuromics and other projects, and we have updated them and for the Solvardi project.
Okay, excellent. So with that, I think we can leave it there. I'm not sure if you, Berta or Leslie, you'd like to say a couple of words, but from here, I'd like to say thank you very much for joining us, and I hope it's been useful.
Yes, I also would like to thank you, Leslie and Ricky and Steve for giving this webinar, and yeah, maybe we see you on Friday for the next webinar on how to analyze your cases with GPAP.
Great, thank you, everybody.
Thank you.
Okay, thank you. Bye.
Bye.